assistant professor from the Shevchenko National University of Kyiv, our collaborator, with a report entitled Circadian Variation in Functional Polarization of Murine Peritoneal Macrophages. Roman, you can share your screen. And please keep 10 to 12 minutes.
Okay, I will try. Okay, so do you see my presentation?
Yes, we have full screen.
Okay, so I'm happy to show you my presentation on a topic entitled Circadian Variation in Functional Polarization of Murine Peritoneal Macrophages. So first of all, circadian rhythm, it's an internal process which regulates many physiological functions such as blood pressure, body temperature, sleep-wake cycles, etc. And these cycles, circadian cycles, they remain roughly 24 hours in duration. And these cycles allow the organism to adapt to repeating constant changes, daily changes in environment, such as, for example, the sunset and sunrise. And it is possible because these circadian rhythms are regulated by circadian clocks. As you can see from the slide, there are master clock and peripheral clocks located in throughout the body in different tissues. Under certain conditions, such as clock gene mutations, shift work, repeated jet lag, sleep restriction, the circadian disruption may happen. And in case of chronic circadian disruption, it may lead to development of different inflammatory conditions, such as cardiovascular diseases, diabetes, obesity, and etc. Our study is focused on macrophages. Macrophages are versatile cells that play many roles. They are plastic and can adapt a variety of activation states, ranging from classically activated pro-inflammatory macrophages to anti-inflammatory alternatively activated macrophages. As you can see, M2 alternatively activated macrophages are further divided into different subtypes, which are activated by different stimuli and perform different functions. So this metabolic polarization into different states helps macrophages to perform different functions depending on a tissue context. But in some cases, the abnormal metabolic skew of macrophages may lead to development of pathologies listed on this slide. Our current study is focused on peritoneal macrophages, which are an important component of omentum-associated lymphoid tissue, and they are involved in pathogenesis of many inflammatory diseases, which makes them promising target for immunotherapy. And despite a lot of research that demonstrated that circadian rhythmicity of macrophage functions, temporal variation in metabolic polarization of peritoneal macrophages remains understudied. Therefore, this study aimed to explore the diurnal variation in the metabolic profile of murine peritoneal macrophages. So we used male outbreed mice aged between 8 to 12 weeks, maintained on 12 hours light, 12 hours dark cycle. About the Z-Geber time scale, I will tell on the next slide. Resident peritoneal macrophages were isolated by flushing the peritoneal cavity with called DPBS, collected at 6 time points at 4-hour regular intervals. The time points are denoted in ZT type, ZT4, ZT8, ZT12, and so on. Macrophages were enriched by adhesion to culture plastic. Different parameters such as phagocytosis of fluorescein isothiocyanate labeled inactivated Staphylococcus aureus reactive oxygen species generation, CD80, CD86, and CD206 expression were estimated by flow cytometry. And that data statistical significance was determined using one-way ANOVA followed by the post-hoc Tukey-Kramer test. The values of P less than 0.05 were considered significant. So now about the time scale. First of all, what is Zeitgeber? Zeitgeber is any environmental cue which entrains and regulates the body rhythms. So the most powerful Zeitgeber, the most powerful timekeeper is the light. And the Zeitgeber time is a scale which is adjusted to light. So it starts at the beginning of the light cycle at ZT0 and finishes at ZT12. And after ZT12 to ZT24 or ZT0, it's the dark phase. It's the artificially maintained cycle in laboratory. And also, it is important to mention that mice are nocturnal animals, so they are active at night, so they are active in dark phase, and their resting phase is in light phase, so like daylight time. So, first of all, we studied phagocytosis percentage and phagocytosis index. Phagocytosis percentage. We didn't observe any statistically significant diurnal variations in phagocytosis percentage. However, phagocytosis index was significantly lower at resting phase at ZT4 in comparison to ZT12, which marks the beginning of the activity phase of mice. Also, we studied reactive oxygen species production. We found that the percentage of reactive oxygen species producing cells was significantly higher in the active phase at ZT12 and ZT20 in comparison to the resting phase at ZT4. And also we observed sharp increase in intracellular ROS production at ZT12 in comparison to the majority of other time points. So ZT12, it marks the beginning of the active phase of animals, and the elevated reactive oxygen species production is the feature of pro-inflammatory macrophages. So this data indicates that peritoneal macrophages are pro-inflammatory activated at the beginning of the activity phase. And these results are corroborated by the findings of other scientists, which detected increase in pro-inflammatory cytokines and chemokines at the beginning, near the onset of organism activity. So it corroborates our data about pro-inflammatory activation of macrophages at ZT12. And conversely, in the resting phase at ZT4, we observed a significantly lower percentage of reactive oxygen species producing cells in comparison to the activity phase, which indicates that macrophages perform regenerative functions in the resting phase. They are anti-inflammatory and perform regenerative functions. So next we studied the CD206 expression. The percentage of CD206 expression, there was no significant changes in the percentage over the daily life cycle. However, we saw the lowest level of CD206 expression at ZT12 in contrast to reactive oxygen species production at this time point. And this further confirms our observation about pro-inflammatory activation of macrophages at ZT12, because CD206 is a marker of anti-inflammatory macrophages, so it is the lowest at ZT12. Next, we have not found any diurnal variability in CD80 expression. Both percentage and expression level were insignificant over the daily light cycle. And the percentage of CD86 expressing cells also did not differ at different time points, but we saw significantly higher CD86 expression level at ZT8 in the resting phase in comparison to the active phase at ZT20. And CD80, which I showed in the previous slide, it is the marker of pro-inflammatory M1 macrophages. And CD86, in contrast, it is regarded as both the marker of pro-inflammatory macrophages, but also the marker of anti-inflammatory M2B subpopulation of macrophages. And therefore, the increase in level of CD86 in parallel with the absence of changes in CD80 expression level indicates that macrophages are anti-inflammatory in the resting phase in comparison to the activity phase. In conclusion, on this picture, you can see the summary of our findings. So in the resting phase, we observed the relative anti-inflammatory shift of peritoneal macrophages, which was manifested by decreased phagocytic index, decreased percentage of reactive oxygen species producing cells, and increased expression levels of CD206 and CD86. Then we observed sharp pro-inflammatory shift of macrophages at the beginning of the mouse activity at ZT12, which was manifested by high, elevated phagocytic index, elevated percentage of reactive oxygen species producing cells, and elevated production of intracellular reactive oxygen species, and decreased CD206 expression. And then in the activity phase, we observed a relative pro-inflammatory shift of macrophages in comparison to resting phase, manifested by increased percentage of reactive oxygen species producing cells and decreased CD86 expression level. I want to thank my team, my scientific teammates, and thank you all for your attention.
Thank you, Roman. Dear colleagues, do you have any questions? I'm from the same team. I can't ask the questions. Please, do you have? No.